All right. Welcome, everybody, to Standing for Truth. My name is Donny Budinsky, and this is Sam. We are your hosts for tonight's exciting program on soft tissue and molecular biology from a creationist perspective. On Standing for Truth, we uphold the truth of biblical creation. And it is a privilege to have Dr. Joel Brown here with us tonight to assist us in accomplishing this important mission. Get ready to have your mind blown by the scientific evidence from soft tissue and molecular biology that confirms the Bible.
Donnie, I can't wait for this. Original organic materials found in the fossils that are claimed to be millions of years old. They provide some of the most incredible evidence for young Earth creation. If you're a critic of biblical creation and you watch this program with an open mind, we pray that you might have your mind changed by the end of this must-watch program.
Amen, brother. That is right, Sam. It is truly an incredible time to be a biblical creationist. And Dr. Joel Brown is going to help prove why tonight. Dr. Brown, it is great to have you for this program. I appreciate you being willing to hang out with us for a couple hours here on such an important topic. Joel, how are you, brother?
Thanks for having me.
Yeah, my pleasure. I'm pumped for this. What a fascinating topic. Truly one of the best of many lines of evidence for young Earth creation. And before we get into your presentation, it'll be a presentation to remember. I can promise the audience that. But before we get into that, Joel, Sam, let's hand it over to you. And will you do the honors of giving our guests here a formal introduction?
No problem. Here we go. So Dr. Brown holds a Ph.D. in genetics, genomics, and development from Cornell University. He holds a BS in biology education from Pensacola Christian College and an MS in biology from the University of West Florida. His academic record is outstanding with a 4.0 GPA in his doctoral studies. During his graduate studies, he researched mouse development genetics and used fruit flies as a model system for studying a protease involved in the onset of Alzheimer's disease in humans. After his doctoral studies, he also went on to complete his post-doctoral studies at Washington University School of Medicine, where he investigated mechanisms of neutral regeneration in the spinal cord of zebrafish, with a potential application for healing nerve damage in mammals. In addition to his research experience, Dr. Brown has significant teaching experience at the secondary and post-secondary levels, including undergraduate, general biology, and genetics labs. In his most recent position, Dr. Brown was the coordinator for the Little Creek Nature Area in St. Louis, Missouri, where he taught biology class to public high school students and managed a 7-person staff and a 97-acre nature area that serves as an outdoor classroom for K through 12 students and provides educational programs to the community.
Sam, appreciate that. Dr. Joel Brown, in other words, you're more than qualified, brother, to be speaking on this topic tonight. Again, for anybody in the audience who is just joining us, that would be soft tissue and molecular biology from a creationist perspective. For anybody who wants to check out the Creation Research website, please do check it out here. You can support CRS there. I've got the links in the live chat and also the description box of this video. And so with that, Dr. Brown, we're going to get right into the fun and hand it over to you for your presentation tonight.
And I will also, I'll share a little bit about myself and the CRS in process of the presentation.
No problem.
All right. Shall I share my screen, Donnie? Are you ready for that?
Yes, we are good to go. As you're getting your screen shared, I'll remind our live audience as people are continuing to make their way in. Once we're done with Joel's presentation here, as we usually do, we do like to leave no stone unturned here on these programs. We prefer to be comprehensive. And so we will be interacting and engaging with your questions, with your challenges. I can see that we got a healthy mix of critics and non-critics already. So please, if you're a critic and you object to anything, or you want to ask a clarifying question, do so in a professional manner, of course, and we will have some fun interacting with your comments and your questions after Joel's presentation here. Joel, I see your slides. A smoldering in the grave. It is up, brother. You're good to go.
Okay. Hey, well, I really appreciate this opportunity. Introduction's way too complimentary. I just sum it up as I'm an ordinary guy that with a God-given love for science, and in my view, that means I love studying creation. Science is the study of how the world works. God made the world, and therefore all science is creation science from my perspective. So that's all I am, is just someone who loves to study how the world works. And this tonight, I'd like to share some of our recent research in this question of soft tissue preservation in dinosaurs. So let's jump in. 66 million years ago, dinosaurs had the ultimate bad day when an asteroid struck planet Earth, bringing the age of dinosaurs to a dramatic close. This is how the story goes. In fact, you see this all over, whether you go to a natural history museum, whether you watch on YouTube, documentaries, whatever. You see this everywhere you turn. How an asteroid ended the age of the dinosaurs. 66 million years ago, dinosaurs had the ultimate bad day. With a devastating asteroid impact, a reign that had lasted 180 million years was abruptly ended. In fact, this is so common that you would have to be just a little bit crazy to believe otherwise. So permit me tonight to be a little bit crazy as we look into some of the actual empirical evidence that suggests that something is not right with this story. As we jump into a smoldering in the grave, let me just introduce myself briefly. Sam, you did a great job. Again, too complimentary, but I spent 13 years in school. I don't know if that's a compliment or a disgrace. I just love learning and enjoyed pursuing graduate studies. I did Alzheimer's disease work in fruit flies. My main thing was embryonic development, and we did some stuff in zebrafish. I'm not here to talk about that tonight, although this is my background, so I love doing it. Molecular biology, tiny stuff you can't see without a very, very expensive microscope. So recently, my family and I'm blessed to have a beautiful family, my wife, and 8 children. We moved here to Arizona just about a year ago, where I took up the position as director of the Creation Research Society. So let me tell you just a little bit about what the Creation Research Society is. It's the oldest and the largest network of creation scientists in the world. We do a number of different things. We publish research. So there's the Creation Research Society Quarterly. This is technical literature on the questions of creation evolution. So if you get tired of reading like the surface level stuff, that's just like an endless repetition of kind of the same stuff over and over again. Even from creation perspective, creationists do that as well. This is going to be technical literature. So check it out. We present research. So we have a conference every year. This is just like the scientific conferences I went to in grad school. So I invite you to check it out. This year it's in St. Louis. It's in July. Creationresearch.org. You can find information about those conferences. Again, this is a technical conference, so it's not just your normal presentations, but a little more technical presentations. We perform research. So this is our lab, the Van Andel Creation Research Center. We're housed on the campus of Arizona Christian University. We have a really nice facility and work primarily with undergrads to perform creation-based research. And we fund research. So I throw this in at the end, but it's an important one. So funding for creation science is not something that's easy to come by, but we will give you some seed money if you've got a great idea. The Creation Research Society wants to help fund those ideas as we seek to better understand the world, but from a biblical creation perspective. So that's what we are, the Creation Research Society. Shameless plug, join the movement. If you haven't, if you're a scientist and a Bible-believing Christian that loves God and loves science, then the Creation Research Society is for you. I only joined it recently, a couple years ago. I went through most of my graduate school wanting something like this, and then I found it. So I joined the society, and now here I am as the director. So I am out there looking for people like myself that were in grad school, that wanted to contribute, that loved science and loved the Bible. And this is how you can do it. So definitely check us out. But to the main topic, smoldering in the grave. So going back to our T-Rex, it's actually surprising. I'm gonna back up there once. It's surprising to think how recently we've actually learned about dinosaurs. You know, we take them for granted, right? They're everywhere in our culture, but this was not always so. You know, it was only within 200 years that we first started to discover these creatures. So the British scientist William Buckland, he coined the term, the name of the first dinosaur, Megalosaurus. So he named the first dinosaur 1824. That was 200 years ago. Before that, dinosaurs, this whole class of organisms was just not even on our radar. I think that's fascinating to think about. Here is the Megalosaurus, the second dinosaur ever named, the iguanodon, by British scientist Gideon Mantle. Coined this name in 1825. Again, just a mere 200 years ago that we just started learning about these things. The word dinosaur wasn't even coined until 1841. Now, over the last 200 years, we have learned an incredible amount. I'm not a paleontologist, so I have enjoyed kind of geeking out over dinosaurs. Who doesn't love dinosaurs? So I had the opportunity just a couple months ago to visit the SWAU Dinosaur Science Museum in Keene, Texas. By the way, it's a fantastic research facility. They have the world's largest collection of geolocated dinosaur fossils. So, and they do this all from a biblical perspective as well. When I say geolocated, here's what I mean. They've been going to the same site for every summer for 20 years, and they have GPS coordinates and 3D scans of every single bone that they've been pulling out of the ground over the course of those 20 years. So it's a huge collection of over 40,000 bones. This is the director, or former director. So they just have tons and tons. Here is a T-Rex bone that I'm holding. There is our T-Rex guy right there. That's the T-Rex tooth. And so I say all that to say we have learned an awful lot about dinosaurs in the last 200 years. I'm going to stop sharing and show you one that we've got right here. Gotta get my special gloves on. So not a paleontologist myself, but I've certainly picked up an interest in dinosaurs. So here is an ulna from a triceratops. So it's huge, it's heavy, it's big, it's solid. Now we have these things by the hundreds of thousands all over the world. So we know an immense amount about dinosaurs, which raised an interesting question. Wow. Let's see if that screen fixes itself. I'm going to share my screen again.
I was going to say, Joel, that's a workout just carrying that thing. That's incredible.
Yeah. Okay, so there we go. Let's talk about this now from a biblical and evolutionary perspective, and some of the differences that lie. So both creationists and evolutionists, they look at the world and they see a pattern of fossils in a geologic column. Yet we have vastly different interpretations of that. So in the evolutionary, the common descent perspective, they see, and when they look at the fossil record, a progression of time, a progression from simple to complex. So when a biblical creationist looks at this same record, we too see a progression we see a progression of the flood. So these organisms represent, for the most part, the geologic column represents organisms which were buried during the flood, including the dinosaurs. So let's ask a simple question. When did the age of dinosaurs end? There's the age of dinosaurs, the Mesozoic era, the Cretaceous, Jurassic, Triassic, sort of in the middle of the column, of the sedimentary column. And we can ask simply, from a biblical perspective, these layers would have been laid down by the flood. All creation scientists agree that the Mesozoic layers were laid down by the flood sediment. And so that would be about 4,400 years ago, thousands of years ago. Compare this to the evolutionary timeline. That would be, they would say the age of the dinosaurs ended with that asteroid impact of 65, 66 million years ago. So here at the start, we see a very big difference between the age of the dinosaurs. Therefore, it came as a surprise initially when in 2005, Mary Schweitzer published a very famous paper describing the presence of soft tissues, vessels, and cellular preservation in the T-Rex. So here's what we're talking about. They were working with the T-Rex bone from the Hell Creek Formation, which is, by the way, the same formation that Triceratops bone that I just showed you. That's the same formation it came from. And they dissolve these bones and they find fibrous tissue, like you see over here on the left. They also found blood vessels, things that looked like blood cells, as you see over here on the right. Now, this was 20 years ago. They actually started publishing this in the late 90s, 1997, some of the first work. And then, of course, it was very controversial. So a lot of work has been done since then by Mary Schweitzer and others to confirm that these are indeed endogenous, meaning they were part of the dinosaur organic tissue, soft tissue. In fact, here at the CRS, we started the iDino project. This was about 15 years ago, before my time, and published several papers studying this soft tissue ourselves. This is the investigation of dinosaur intact natural osteo tissues. So here's an example of some work that was done here at CRS. And this is, again, the Hell Creek Formation in Montana, same place that Mary Schweitzer's T-Rex bones came from. So there was some scientists from CRS that went up there and got some bones themselves, specifically this Triceratops horn. And this was a 46.5-inch brow horn encased in sandstone. Took it back to the lab. And they did a number of different studies. Much like Schweitzer, they can do thin sections of these bones and look at the ultra structure of the bones. So what you see in this one is thin sections, and you have these cells complete with filopodia. These are osteocytes. They're bone cells within the horn, and beautiful morphology too, I might add. So these cells have been preserved that long. That doesn't mean the cell is alive. Now, let's be clear, the cell's not alive, but these are the skeleton of the cell and some of the structures that remain. Here is SEM, again, taken by scientists that were working with CRS. So electron micrographs showing these bone cells along with their filopodia. It's actually amazing to see this level of preservation. This surprised even creationists, right? You know, this thing's been buried for thousands of years. It's amazing that we're finding this type of material. Here's another bone that they were working on. It's from a Thescelosaurus, that's vertebra, so part of the backbone. And this is a video taken here at our lab. They took the bone, they took a piece of this vertebra, and they dissolved it. So when they dissolve it, what they're doing, they're removing the mineral component. And what's left is anything that's soft. So here's what was left. So rather than being just completely dried out and with no organic substance at all, there's actually tissue left in the bone. So this is from after the bone is dissolved, there's soft tissue that is still remaining. Here is a little vessel, presumably a blood vessel, coming from this dinosaur bone, still flexible, still pliable. Again, this is not, at this point in 2012 when this work was done, it was no longer surprising to find this type of material, because Schweitzer's lab and others had been finding this now for years. In fact, there have been over 100 papers reporting soft tissue in dinosaur and other paleontological specimens. So this is a well-known phenomenon at this point. There's still some people that argue that those are exogenous tissues, so they weren't part of the original dinosaur that remains. But by and large, even in the secular community, this is well accepted that there's these soft organic tissues. And the reason why is because Mary Schweitzer, let's give her credit. She has gone above and beyond to demonstrate. So over 100 papers, they find pliable tissue in a variety of dinosaur bones, morphologically distinct cells. Collagen fragments are also frequently detected, and other proteins, actin, myosin, tubulin. These are all detected now. The proteins like those are vertebrate-specific proteins. So here is a very recent one. So we've been talking about stuff 20 years ago. Here's perhaps the most recent one. Published in 2025, evidence for endogenous collagen in Edmontosaurus fossil bones. So in this paper, they actually sequenced the collagen proteins that were found in this bone. So that was one of the really novel additions that this paper did. It wasn't the first time a dinosaur protein had been sequenced, but it corroborated some of Schweitzer's results. They also do this imaging to show the pink represents the presence of a structured crystals and collagen in the bones. So that's this is 2025 now, and we're still seeing it. Now, I want to direct your attention towards that word collagen. We're going to spend the rest of our time talking about that very important protein, collagen. Maybe you've heard about it. Maybe you heard about it this past week when you saw some headline, some clickbait headline come across your screen. World's first T-Rex leather is claimed to come from dino DNA. Is this for real? So I've started seeing these headlines pop up all over. This was a news blurb, a news release from a company. They're trying to make synthetic leather. When I say synthetic, though, lab grown leather. So instead of killing a cow and getting its hide and making leather, you could grow that leather by growing cells inside of a laboratory. Well, this company took it one step further, and they are trying to do this from dinosaur proteins. Now, in this article, they say a blueprint for this lab-grown leather is fossilized T-Rex collagen. There's that word. I want you to focus on collagen. It's gonna be the main character in our plot. So there you have it. There is collagen is being found routinely in dinosaur bones, to the point where we can now sequence the collagen and back calculate what the DNA sequence was for that collagen. Then scientists could then take that DNA sequence, grow that collagen in cell culture, and make a T-Rex leather purse. At least that's what they claim. The T-Rex purse might be a stretch. We'll see. So anyways, that's what we're talking about. What is collagen? Let's start there. Well, I'm so glad you asked. So I have a couple of visual aids that will help us. You might be thinking of some sort of anti-aging cream when you hear about collagen. You see this all the time on supplements. You might mix it into your smoothies, collagen. Well, what in the world are they talking about? What's this word collagen that keeps coming up? Well, simply put, collagen is just a string of amino acids. I'm going to demonstrate this live. So I have a little prop. Here is a single amino acid. This is actually the simplest amino acid. It's glycine. All right, so a protein is just a chain of amino acids. But collagen has a very specific arrangement, just like all proteins. So let me show you a little bit about that arrangement. I'm going to take, you can combine these together, by the way, by removing some water. It's a dehydration reaction, and you can combine amino acids together. So here's another one. This is alanine. Here's my glycine, and they come together. Now we have 2 amino acids. Now I want to point out this purple bond. This purple bond is called the peptide bond. That's the bond that connects the 2 amino acids. That's going to factor very large in the rest of this talk. So keep an eye on those purple bonds. Let's add one more amino acid, shall we? Now these collagen is hundreds of amino acids long, so we don't have time to build all of that. But let's at least build these 3. So I just added on a kind of chubby amino acid. This is called proline. And it's, well, I won't go into the details of why it's kind of a unique one, but it has a shape that's different from the other amino acids. So here we have these 3 amino acids, glycine, alanine, and proline. This forms the foundation of collagen. Let me show you what I mean by that.
I'm loving those props, Joel. Very good.
Yeah, so we're going to see kind of how I just showed you 3 amino acids. Let's see how they fit into the overall structure. So here is the structure of collagen. Collagen is just a protein. It's a chain of amino acids. Remember all of those purple bonds I showed you 2? Those were the peptide bonds. And I've marked those with the red dotted lines here. Keep an eye on those peptide bonds. The whole rest of the presentation really hinges on that. So here is the full sequence. I just showed you 3 amino acids. This is the full sequence for type 1 collagen from cow. And I want you to look at this very closely. I want you to try to find a pattern. Do you see any pattern in this sequence? I'm going to give you some wait time. I'm not expecting a response, but if you know it, put it in the chat. I'm going to help by zooming in on it a little bit. Look very closely. Do you see a pattern? Okay. Hopefully you've had a little chance to notice. You see every third letter is a G. In fact, there's the GAP. Those are the 3 amino acids that I put together for you. The glycine, alanine, and the proline. So, but every third letter is a glycine. That's a very specific organization in collagen, and it gives it a very specific shape. So here's what collagen looks like inside of your body. By the way, your body is loaded with collagen. It's the most abundant protein in your body, which is why, they put it in all these youthful creams because they think it's somehow going to make you young again. Most abundant protein in your body. And in your body, it's not just one chain of amino acids. It's actually 3 chains that are combined together. So in this diagram, what you're looking at, the red, white, and blue, those are the 3 amino acids that I assembled for you live. And then the other chains are the full 3 separate collagen molecules that twist together to form this higher level structure of collagen. Now, of course, you can't see this with a microscope. These are crystallography, X-ray crystallography, and other techniques are used to solve these protein structures. So that's what the overall structure of collagen looks like. And I say all this to say collagen is extremely stable. It's designed, from my perspective, here's a little worldview input. It's designed to be extremely stable. It's in your bone. It's in your skin. When it ceases to be stable, you get wrinkles, right? So it's designed to be an extremely stable structural molecule. That's what collagen is. So what does collagen have to do with bone structure? I'm so glad you asked. So bone is a mixture of a couple of different things. It's a mixture of minerals, calcium, phosphorus. Those are things that you think about when you think of bone, but perhaps you don't think about the proteins. So it's composed of minerals, but laced throughout that mineral component are the proteins. And here is the collagen matrix. So it's just loaded. Here's a little breakdown. Your bones are about 60% bioapatite, that's the mineral component, about 10% water, and about 30% organics. Out of those organics, of course, include collagen. In fact, collagen 1 is the most abundant protein in the bone. So 27% of your bone is collagen, which is crazy to think about, right? Because you don't think of a protein as being such a large component of the bone, but that's why it matters. So this now raises an interesting question. Collagen is in bone. Bone dies. How long then can collagen lie a smoldering in the grave? And that is our topic. So I want to, again, draw your attention to those peptide bonds, because it turns out you can use these peptide bonds to determine how long that collagen is lasting. Keep in mind, when we say, how long does that collagen last? Here's what we mean. How long do those peptide bonds last? When something dies, when something begins to decay, all that has to happen is a little bit of heat and a little bit of water, and those bonds will spontaneously begin to break. Those peptide bonds will spontaneously dissolve over time. So how can we measure that? Well, in our lab, we measure that using Fourier transformed infrared spectroscopy. What in the world is that? This is a very, very expensive flashlight. That's all. All right, so it's a fancy souped up flashlight. And one of the ways that it's souped up is that it shines an infrared beam of light. So here is our protein, our amino acid sequence, collagen, for instance. And in this case, the blue bond is the peptide bond. And so this region is the same in all proteins. They've all got this red box area. It's also called the amide bond. And you can detect the presence of that bond using infrared. So we shine our very, very expensive flashlight at this amide bond, and it is going to transmit light. So it's going to absorb some of the light and it's going to transmit some of the light. And you can tell exactly which bonds are present in your sample based on what's transmitted. It creates these mountain peaks and a very distinct pattern of mountain peaks depending on what is in your sample. So this is not our work. This is Skagian et al. 2024. So you can see what they're looking at. They're looking in the red line is poorly preserved bone. The green line is well-preserved bone. And they're scanning it with this FTIR, with this infrared laser. And here's the specific peaks that show up. So if there is a lot of this amide 1, if there's a lot of protein, a lot of collagen present, then you're going to have a tall mountain peak here, which you see in the well-preserved bone. But if there's not a lot, then you'll see a reduced mountain peak, which you see in the poorly preserved bone. And you notice what doesn't change. So direct your attention to this phosphate peak. That's what that PO4 means, phosphate. That's the mineral component of the bone. So that's representing that shouldn't change over time, but the amide 1 peptide bond should. And indeed, here's another paper that's doing this with FTIR. Here's they're doing a artificial decay of bone. So at time 0, here's the mountain peak. And this peak is very specific. It's at a very specific wavelength. And at day 14, that peak has been reduced. Okay, this is very important. We can now use this. We can exploit this characteristic of bone and bone collagen to measure decay over time. So here is stuff from our lab. I'm showing you 3 pink lines. These are the absorption spectra of different bones at different time points. Let's zoom in. So the blue line represents a bone at time 0, at the beginning of the experiment. I want you to focus on 2 peaks. The amide 1 peak, that is those peptide bonds. That's the purple bond. That's what we care about. And then the phosphate peak over here, that's the mineral component that doesn't change. And so what you see is that at time 0, the blue line, we've got a nice strong peak. 7 days later, we have a reduced peak. 14 days later, the peak gets keeps getting smaller. So we can do this artificial decay process in collagen bones, or in bones, to study the decay of collagen. Now we accelerate the decay because this is an extremely stable protein, and we're not going to wait around hundreds of years to get our results. So we accelerate the decay. All right, so let's jump into the science. We are just simply examining the collagen decay in these different organisms. We chose 3 different taxa. So we're studying this in cow bone, we're studying this in ostrich bone, and alligator bone. This is what it looks like to get bones from alligators. It was probably the only time I will ever do that in my life. I never expected when I got my PhD in genetics that I would be dissecting alligator legs. So there we are, getting the bones from the alligators. And of course, we do this for the ostrich, for the cow, and we get our bone samples. Here is how this experiment is set up, our materials and methods. So we're starting with the bone. We then crush this into bone fragments that are on the order of about 1 millimeter in length. We incubate these at a range of temperatures, and we do it over a course of time. We then pull the bone after it's been cooked and we grind it to a very fine powder. We measure this to 20 to 63 microns. That's millionths of a meter. We scan that powder. This is where the FTIR comes in, and that generates these peaks. We are specifically looking at those 2 peaks, the phosphate and the amide 1 peak. We then use this to build decay curves. So we're doing this at different temperatures. So we should see the collagen decay over time, and we generate these different decay curves at the different temperatures. So you can then use the decay curves to build something called an Arrhenius plot. The Arrhenius plot is it's just a plot showing the relationship between the decay and temperature. As temperature increases, decay increases, and so you can build this a relationship between decay rate and temperature. Now, here's why we're doing this. We can estimate at what temperature these dinosaurs would have been fossilized in, the ground temperature where fossilization occurs. So we can generate decay rates here in the lab, and then calculate what those would be at the physiological temperatures of decay. Now, here's the thing. We are only testing one factor in decay, and that is the thermal decay rate, meaning how much energy, thermal energy, is required to break these bonds. There are many other factors that are involved in decay, such as bacterial degradation. If there's bacteria, then they actually start chewing up the peptide bonds themselves. So there are other factors involved, but this is just a theoretical maximum for the decay of collagen, the thermal decay. All right, here's the result. Now, I'm going to walk you through this, but I wanted to throw this at you. And I want you to first realize and appreciate each point on each of these curves, which I will explain in just a moment. Each of these points represents an experiment that's 2 weeks long, that has 5 different samples, that represents hours of grinding these bones. So there's an enormous amount of work that goes into generating this data. And I wanted to give some credit to our lab. I'm going to play just a short video showing this overall process.
Dr. Brown, is this supposed to have sound? Here we go. You can hear it now.
It's just coming through the speakers. I'm gonna just turn that down, and I'm gonna narrate something. There is no narration. It's just a visual. But since you're not hearing the sound, I'm just gonna narrate what you're seeing. Okay, so what you've seen so far is, we've ground the bone. We have now these homogeneous samples. And now comes the cooking part. Now, when I say cooking, kind of colloquially, what we're doing is we're adding thermal energy. And this is because it's known that the collagen is going to last for hundreds of years. And nobody's going to do an experiment that lasts hundreds of years. So when you add more thermal energy, it speeds up the decay process, which is okay, because what we're doing is we're just calculating at each temperature what's the decay rate, and we'll use that to study the decay rate at physiological temperatures. So here comes, after the bone has been cooked, we then have to go, this is the hardest part, go through the painstaking process of grinding it. And this is 3 samples, but of course, in reality, this is done with dozens and dozens and dozens and dozens of samples. Really gives you a good forearm workout. So we still grind the old-fashioned way, just a good old mortar and pestle. It takes a lot longer than what that video just implied. So what you're seeing here is now it's a powder. The bone has been powderized, and we have to select a very specific size in order to do the FTIR analysis. So we are selecting just the powder that's between 20 and 63 microns in diameter. There it is. Once we've reached this stage, we are then ready to pass it to the next person. Now it's time to measure using the FTIR. So it's a powder, but inside of that powder, there's both the mineral component and there is protein. There's collagen. So the FTIR is a lot scarier than it sounds. It's actually like super straightforward. You place your sample across this window. It shines the beam through there, and it measures how much infrared light is transmitted and absorbed. You get these spectra like so. We do replicates of everything that we test. So there's triplicates in all these experiments. You do this over and over and over again, and you build these giant spreadsheets like so. These are all of the different measurements and averages and all that good stuff. We then use a statistical program called R. It's a phenomenal, free, powerful statistical software. I highly recommend it. You can use it to do your stats and to graph it. So we analyze this data using R, and voila, there is the plot. And these are the plots now that I am showing you. So we are back. Just to make sure we got that transition right. Donnie, can you still hear me?
Yes, Joel, you're
Awesome.
coming in great.
Okay. So I just wanted you to appreciate that when you see just these graph like this, this graph takes about 30 seconds to look at. It takes about 8 months to make. So kudos to all of the folks, undergrads and techs that have helped on this experiment. So now let me back up and explain what we're looking at. All right, so here is one example from one organism, ostrich, at one temperature, 85 degrees Celsius. We're looking at collagen concentration on the y-axis, and we're looking at time on the x-axis. So we're looking at the decay of collagen over time at 85 degrees Celsius in ostrich bone. And you see a nice line there. Right, but we can't do this just at one temperature. We have to do it at a range of temperatures in order to see how the decay rate changes based on temperature. So we do this at 70, 75, 80, 85, and 90. And you see how it's pretty flat at 70 degrees Celsius. At 90, it's pretty steep. So that's, as you would expect, the decay increases. The rate of decay increases with temperature. But we're not doing this just in ostrich. We're doing this in 3 organisms: alligator, cow, and ostrich. And that's how we then get this whole plot at each temperature, each organism. Now, I want to just zoom in on the cow here. So we can then take the decay rate from each of these 5 temperatures, and we use that to build our Arrhenius plot. So I wanted to give you kind of the back end of this so you'd see how these things come. Here is the Arrhenius plot. So this is now plotting on the y-axis the collagen decay rate, on the x-axis the temperature. These points here, those are the decay rates that we empirically measured at 85 degrees Celsius. These points down here are the decay rates that we measured at 75 degrees Celsius. So you can see how the rate of decay increases with temperature. Now here's the important thing. We did all of that work to get that number that is in the red box. That is the slope of this line. And we can use that slope to calculate something called the activation energy. This is the activation energy of the decay of those peptide bonds. That's just a chemical reaction. It's the addition of water. It's called hydrolysis, the addition of water to break the peptide bonds. That's a chemical reaction with an activation energy that can be measured. And you can calculate this by taking that slope that we worked so hard to get. There's the activation energy. This is the ideal gas constant. You plug these items in, and our activation energy is 161,000, give or take, joules per mole Kelvin. Now, if you haven't got anything else from that, this is what you need to get. You can use the activation energy to calculate how long this is going to last. So half-life of collagen at 100 degrees Celsius is 0.71 days. That's very, very short. But of course, life doesn't happen at 100 degrees Celsius. Half-life at 75 degrees Celsius is 32 days, significantly longer. Half-life at 50 degrees Celsius is 7 years, even longer. Half-life at 25 degrees Celsius, 1,175 years. Half-life at 10 degrees Celsius, 39,213 years. So you see how it increases, or the half-life increases as the temperature decreases. Now, I want you to especially focus on that 10 degrees Celsius time point. I'll come back to that in just a sec. So, in summary, we can do this for all of these different organisms. The half-life for alligator is 885 years. The half-life for cow is 1,175, and ostrich comes in at the lowest. Their bone collagen doesn't survive very long, 327 years. This is all at 25 degrees Celsius in a neutral aqueous environment. So we were doing this in a neutral pH 7 water environment. All right. Now, we are not the only people to test this sort of thing. So here's a study from a secular lab, and they also, using different methods, studied how long collagen ought to survive. And here was their conclusion. Extrapolation from high temperature experimental decomposition rates suggests that at a constant 10 degrees Celsius, it will take between 0.2 and 0.7 million years at 10 degrees Celsius for levels of collagen to fall to 1%. It takes 7 half-lives. You start at 100%, 1 half-life later, 50%, 2 half-lives later, 25%, so on and so forth. To fall to less than 1%, that's 7 half-lives. And these are the data that they calculated. Here's our data, just for sake of comparison. We calculated a half-life of 39,213 years at 10 degrees Celsius. This comes out 7 half-lives. That means in approximately, give or take, 250,000 years, the levels of collagen would fall below 1%. So What does that mean? This is very similar to what they found, which is the way science is supposed to work. It's repeatable. So our data lines up with their data. The longest we can reasonably expect collagen to last under these conditions is 250,000 years. In their paper, it was 0.2 to 0.7 million years. Now, this does not tell us how long that collagen has been there. It tells us how long the max that we could expect it to be there under these conditions. Now, in reality, we would expect it to decay faster because in nature, you've got things like bacteria that are contributing to decay. This is just looking at thermal decay. So, to tie this all together, how long can collagen lie a smoldering in the grave? Not 65 million years by conservative empirical estimates. So in order to do this research, we didn't need to appeal to any worldview perspective of our own or of a secular evolutionary worldview perspective. This is just laboratory research, which you could do in your garage if you had that very expensive flashlight. So what do you do with this? This is a good question. It's one that has perplexed secular paleontologists as well. The persistence of still soft vessels, matrix, and cells in fossil remains millions of years old is unexpected. And if original, refutes models of, I wonder what goes in the blank. So herein lies the conundrum. Are they going to say this refutes the models of the age of these bones? No, certainly not. They would say this refutes models of degradation. So in other words, we just don't understand how these things are degrading. Now, at this is just a simple biochemical process. What we have measured, what the other paper has measured, that limits it to a max age of about 500,000 years. So there may be other factors involved that we have yet to test. And here are some of the ideas that are suggested. The identification of still soft tissues and cellular structures in a suite of Mesozoic fossils and claims of endogenous proteins preserved within these materials is controversial because it challenges both conventional wisdom and theoretical kinetics, which preclude the persistence of proteins over geological timescales. So this is a tough problem to get around because these are just empirical measurements that we and others are doing, which say the proteins don't last that long. So here are some of the ideas that secular paleontologists have come up with to try to explain the persistence of these proteins. The cross-linking hypothesis. This is the main one. And it looks something like this. So you would have these radicals that form. And that little beads on a string that represents the collagen. These radical form. And these will come together and they will cross-link. They will covalently bond between these different parts of the chain. That's what cross-linking is. And the idea is that this would stabilize these the collagen molecules and preserve them for longer. Increase resistance to enzymatic and microbial degradation. And sounds great. So the only problem is some people have started testing this. We are in the process of testing these different hypotheses as well. I welcome that they are presenting these hypotheses because that's something that can be tested, right? We can now test this with our model, our system, and see if it actually preserves it. But somebody else tested this in 2023, and they tested a whole bunch of different groundwater chemistries and measured what happened in those bones. And here's what they found. The presence of iron in their experiment actually accelerated decay in this test. So now there's always going to be another hypothesis out there. This is the nature of a historical science. Paleontology, evolution in general is a historical science. And so it's the nature of a historical science. There's always going to be a hypothesis that's a rescue device to work around these. And so we can play whack-a-mole and keep testing those, and we will. But let's not lose sight of the bigger picture here. The struggle. If these components are demonstrated chemically to be original, what would that mean? I can think of something, but here's what Mary Schweitzer says. A mechanism must exist to allow their persistence across geological time. When you see words like that, must exist, what you're seeing is worldview or a faith system in action to be able to explain these devices. So, in summary, when did the age of dinosaurs end? Well, the presence of dinosaur biomaterial is not consistent with the conventional assigned ages of the fossils. The presence of dinosaur biomaterial is consistent with the biblically assigned ages of the fossils. True science affirms the Bible, and it'll be interesting to see the continued set of rescue devices that are employed to get around these empirical data. So I just want to throw this in at the end. If there are proteins, what about DNA? Oh, I'm so glad you've asked. So we can sequence DNA from ancient specimens. DNA here sequenced from hominin fossils, which are 430,000 years old. I put that in quotes. That's the conventional age assigned to these. We would put these post-flood. So they're getting DNA from things that are 430,000 years old in the conventional time scale. DNA sequenced from mammoths, which are 1.1 million years old in the conventional timeline. So it makes you wonder, could we get DNA from dinosaur bones? Now, this is an important difference between the biblical and the evolutionary worldviews. You remember our T-Rex leather? Well, in this article, they point out the oldest DNA fragments on record extracted from Siberian mammoth molars are around 1.6 million years old. T-Rex went extinct 66 million years ago. So there's no hope of ever recovering its DNA. And you hear this from scientists a lot, kind of put a reality check on the popular culture. Dinosaurs died out 66 million years ago. There's no way we're ever going to get DNA from them. Well, if you go back to our timelines, the biblical worldview would predict that if we can get DNA from things that were thousands of years ago, then we should be able to get DNA from things that were killed and laid down as fossils during the flood. Not all of them. I don't want to trivialize this. I don't want to say this is going to be easy, but I do want to stick my neck out there and make the prediction that dinosaur DNA is in a fossil out there somewhere. And so one of our next objectives is going to be to find it. It's not just us, but there's a very talented group of other Bible-believing creation scientists that are interested in this same project in finding that dinosaur DNA. So just to let you know what's on the horizon. True science affirms the Bible. So when we look at the world around us, this is what motivates me. I'm unashamedly hold to a biblical worldview, although the science I presented here is independent of any worldview. The Bible says, "Thou art worthy, O Lord, to receive glory and honor and power, for thou hast created all things, and for thy pleasure they are and were created." Our objective, my personal objective, as a lover of science and lover of God, is to just give credit to whom credit is due. I do want to thank our team here at the Van Andel Creation Research Center. That's the lab at the Creation Research Society. They've put in a lot of hard work on this project and others, and we've just had a great time doing it over the last 8 months. With that, I'm happy to take some questions.
Dr. Joel Brown, that was an incredible presentation, and I made a lot of notes. There's a lot to discuss there. One thing that you said near the end about your prediction on DNA, dinosaur DNA, and that's something we don't yet know, but based on the biblical worldview, I like what you said. Good science or true science always affirms scripture or the Bible. That's so true, brother. But the fact that you're meeting the gold standard of science, making future testable predictions, one of them being DNA in a dinosaur fossil somewhere out there.
Yeah, I that is the fun part of science, and it's actually rare. Oops, sorry. It's actually rare to find such a stark area where there is a such a stark difference in the predictions of the two worldviews. So the evolutionary worldview, They say hands down, and I don't blame them, no chance of DNA surviving. DNA is far less stable than proteins. So the proteins, they're okay trying to find a workaround. The DNA, no chance of that. In the biblical worldview, adding another 1,000 years on to the stability of DNA is totally reasonable. And so it is a prediction that we would find DNA in these fossils.
And we can make another prediction: is should we find that DNA, then they will again wave it away, just like the soft tissue.
That's right.
That's a good point.
Don't hold your breath.
That's right.
Yeah, that's a good prediction. Predicting ahead of time when we find the DNA in a dinosaur fossil, how they would respond. Dr. Jason Lyle did something similar with the James Webb Space Telescope data. And so it's not like they're just going to tap out. They will come up with a rescue device. And Joel, I appreciate how you covered a couple of those rescue devices. And so I'll just start the Q&A off with a bang. And perhaps you can elaborate on this a little bit or give us an elevator pitch response, because I've seen, as I like to do when it comes to these lines of evidence, Joel, that just point so overwhelmingly to the truth of young Earth creation. I like to see, what are the old earthers, what are the theistic evolutionists, the naturalistic evolutionists, what are they saying in response to this? Because as we know, there's a lot of data points that are agnostic to the debate, where both creation and evolution can account for the data. Maybe there's a good explanation, something that's valid, reasonable. Okay. So in a way, there's certain pieces of evidence that are consistent with both models. But when it comes to this soft tissue and blood vessels, red blood cells, bone cells, protein fragments like collagen, as you're pointing to, Joel, these points of evidence seem to be much more consistent with young Earth creation and may even preclude the billions of years paradigm. And so you touched on it briefly. I've seen in the literature prominent old Earth creationists, for example, they will say, well, when it comes to collagen is extremely durable. It's highly intertwined. It's cross-linked. You talked about the cross-linking hypothesis. And so this durable nature of collagen essentially gives it the ability to survive for eons. Some have even said eons. And so I'm wondering if you're confronted by a skeptic, maybe you're on an elevator pitch, and they bring that up and they're a reasonable person, they're just looking for a good response to that, I think, rescue device, what would be the best way to engage that, Joel?
Yeah. Okay. So now you're speaking my language, because when we're talking about historical science, it is incredibly frustrating. It's like playing whack-a-mole to try to deal with every idea that comes up. It's impossible, in fact.
Right.
When somebody says that, I like to say, okay, well, let's test that. That's our whole purpose at CRS of having a lab, is let's test that. And let's not test it with a theoretical protein. Let's test it with collagen. Let's not just test it with collagen, free collagen. Let's test it with bone collagen. And so we can perform those tests and let the data speak for itself. So I would tell them, yeah, funny thing is, we just did that very test you're describing. And we show that 25 degrees Celsius, it lasts about 1,000 years. At 10 degrees Celsius, it lasts about 35,000 years to 250,000 years when it starts. So yeah, based on that data, it doesn't survive. It is stable. It's designed to be stable, but it's not 66 million years stable based on empirical data.
Wow. So when a prominent anti-young Earth creationist or old Earth creationist says, well, in a nutshell, we can get these incredible preservations that, from my understanding, weren't predicted beforehand. And so the way I see a lot of these explanations is they're largely post hoc, ad hoc rationalizations. And so when they say, well, okay, in a nutshell, we can explain these preservations due to the durability, but also, and so perhaps you can touch on this, one prominent old Earth creationist from Reasons to Believe said, the abundances of proteins in fossilized remains of dinosaurs because there's so many of them, because they're in abundance, of course some are going to be preserved, maybe just by a matter of chance. I'd have to see a person like that elaborate on that point. But have you heard that, Joel? And what's a good response?
Yeah, I mean, so half-lives don't care that much about abundance, right? Half-life is independent of abundance. So no matter how much you start with, you're going to end up with 1% of that after 7 half-lives. And so when we're sampling these bones, we're not taking the entire dinosaur bone, or the people that find the soft tissue, the people that measure the collagen, they're not taking the entire dinosaur bone and pulling a tiny little piece of collagen out of it. They're taking a very small piece of that dinosaur bone, and they're able to extract collagen out of it. So there's no reason why dinosaur bones should have a higher concentration of collagen. That's quite a claim if you think dinosaur bones have a higher concentration of collagen compared to modern bones in these different taxa.
Does it sound like to you, and then Sam, I want to throw it to you for a question, brother. Does it sound like to you a lot of these rescue mechanisms or so-called explanations, they are driven by the evolutionary or old Earth presuppositions that a lot of these people have, because they have that worldview, the old Earth worldview. They're confronted with this data, these biomolecules or organic material that's found where it shouldn't be. And so rather than tapping out their worldview that they're not willing to change or give up, it's driving those conclusions. I guess for you, Joel, I'm curious what your thoughts are on that, because if they're just appealing to these rescue devices rather than conceding what I think is the obvious point, is it because they have a basic worldview that's driving those conclusions?
Yeah. So I started kind of getting at that and pointing that out with some of those quotes at the end, where when confronted with this evidence, in Mary Schweitzer's quote, she says, if these are determined to be endogenous organic material, then they must, and then you wonder, okay, what's she going to come up with? Then there must be a degradation mechanism that we're not aware of that allows preservation for deep time. Okay, is that the only possibility you can think of? I can think of another one, and that is that the bones are not that old. But when people are using that language, then that shows, without them often even aware, it shows that they are committed to a worldview, and unable to consider alternatives outside that worldview. And to be fair, creationists do the same thing, right? This is historical science that we're talking about. This is not empirical science. The stuff we did in the lab was empirical science. That's I wouldn't want to have to argue against that. But the historical side, when you're talking about how things happen in the past, that's outside of the realm of testable science. Evolution, as you're well aware, is not an empirical science. It's a historical science. Creation science is not empirical science. It's a historical science in that regard. And so both sides do seek to find explanations. I think you have to do some enormous gymnastics to explain your way out of this one.
Right. I think you made a lot of good points there. And I know, Sam, you'd love to touch on this too, because we all have worldviews that we're coming to the data with. And so both sides, creation and evolution, are going to be faced, I think, with pieces of evidence or data points where we are going to have to come up with an explanation.
Yep.
Maybe even a rescue device, right? Probably both sides are going to have a rescue device here and there. And so I think when I'm looking at these explanations, I'm thinking to myself, okay, are they reasonable? Are they consistent? Or do they largely comprise imagination? Because again, I've seen strong points thrown at the evolutionists, but then they come up with an explanation that might not even be true, but okay, it's reasonable. Maybe that's just an agnostic data point. But again, coming to this issue of soft tissue and collagen and so on and so forth, I'm not seeing a real good, reasonable, or convincing rescue device presented.
And as we've mentioned, the most common one you're aware of, and it's the cross-linking hypothesis.
Yeah.
And the idea being, of course, there iron within the blood of these deceased organisms. As the red blood cells break apart, the iron mixes with collagen, causes these cross-linkings. And people are going to fight about whether that's reasonable or not all day long. I hate those fights. I'd rather go to the lab and say, okay, let's
Yes.
test it.
Yeah.
And so we are.
I love that approach, Joel. You're doing an excellent job. Sam, I'll hand it to you, brother. You got a question?
Yeah, this question comes in from the boys that I ride Harleys with. We want to know where we can buy the T-Rex leather jackets.
Yeah, well,
Just kidding.
right now it's just speculative investment. So
That's right. But I do have a
Well,
question.
hey, Sam, before you do, I was teasing the chat earlier because we always get a good mix, Joel, of critics and non-critics. And I enjoy that. It's fun. That way it's not just purely an echo chamber. We can see what they have to say. Hopefully we can plant some seeds because it really is about winning souls. We could win arguments all day, brothers, but it's about winning soul. He that winneth souls is wise, right? And so we do want to plant some seeds and maybe tease our evolutionist friends a little bit. And so, yeah, I pointed out that a T-Rex lab-made collagen purse or handbag or whatever wouldn't be possible if the earth were billions of years old. And so that was the first time I heard that one, Joel. So that's pretty fascinating, brother.
yeah, this is fresh off the press.
Yeah, that is cool.
Go ahead, brother. Over to you.
So I have a question here, and this is regarding Dr. Fuzz Rana, and he's argued that a chemically altered form of collagen will persist in animal remains well beyond millions of years, especially if the creatures are large like dinosaurs. And so can you talk a little bit about that, and how would you respond to that?
Yeah, so there is I assume the chemical modification they're talking about is the cross-linking. It also goes back to that cross-linking. What they're saying is that you have these modifications that then increase the stability. So to address that, you have to be able to provide a reasonable mechanism for the iron or for whatever cross-linker you want to propose. So you have to propose that inside that deceased animal, there is a chemical that in modern chemistry can be used to cross-link. It has to be present at high enough concentrations to produce the cross-linkings that are necessary. And then you have to demonstrate that cross-linking actually preserves the bone. So the burden of proof is on them. They're welcome to present that evidence. Like I said, those who have tested the iron decay, usually irons the cross-linker. Their 2023 paper actually accelerated the decay.
Yeah, I was going to ask that, Sam, maybe just elaborate on that, because in my notes, closer to the end, you said there was a 2023 test, which you just reiterated, where iron actually sped up the decay process. So therefore, isn't iron doing the opposite of what they want iron to do?
Yeah. So they did not. Now in that paper, they did not check like the level of cross-linking. You can measure how much cross-linking has occurred inside the bone and such. So they didn't test the level of cross-linking, but they tested iron specifically because that was the primary mechanism that's been proposed. And so they don't know why it accelerated the decay. That's just the nature of empirical science. Sometimes you just have the data. The data says it actually survived less well in the presence of iron.
Do they have anything else? They must be hopeful. There must be something else now that iron is a problem.
Well, so there's another idea out there called glycation, where you have a sugar. So this is based on not the iron in the blood, but blood sugar. And the sugar, it becomes a reducing sugar. It binds to the collagen, and it again chemically modifies it to preserve it from degradation. And when they say degradation, there are several different types of degradation. There's bacterial degradation. We have not even considered that. That would only decrease the number. We are only looking at thermal degradation, which is a simple chemical reaction in which water goes in and breaks the bond. So they're saying that, well, if you've got the sugar that's bound to collagen, now it's blocking the water and it will no longer break that bond. So we're testing both of those, testing it using iron, testing it using hemoglobin, testing it using sugar to test those hypotheses.
Joel, in relation to the 2023 paper, what if someone were to say, and we do have someone in the chat saying it, that the iron was sped up, the iron decay is sped up the decay process because it was, well, a highly acidic condition, so a very unique condition in this scenario. Joel, what's a good response to that?
So they're saying like in the reason the iron sped it up in the 2023 paper.
Yeah, I believe so.
Yeah, so they're trying to test these things at reasonable levels. So that paper was testing groundwater. And so they weren't just spiking this with unrealistic levels of iron. They were using levels of iron that would be expected in groundwater. So if it affected the pH in their experiment, it's affecting the pH in the groundwater too.
Yeah, it would have to be a real world scenario, or else what's the point? I would think, right? I mean, if they're gonna posit that it's gonna preserve these, this soft tissue, then they're gonna have to present something that would be, I guess, plausible in the environment.
Yeah, and so you can demonstrate and show that cross-linking does occur. So this is right now why that's the most popular one. You can see that cross-linking is more prevalent in old samples, and you can treat the samples and cause cross-linking to occur. But the presence of cross-linking does not then equal a from 1 million to 65 million increase in preservation.
Right.
And
right.
that's what remains to be demonstrated. I mean, it's a welcomed hypothesis. I have no qualms with people hypothesizing these things. Let's just call it what it is, a hypothesis that remains to be tested.
Well, because you're doing the testing, and from my understanding, when it comes to your presentation, fantastic presentation. We also had our conference last month. Dr. Brian Thomas gave another incredible presentation. And from what I'm understanding from both, you both pointed to these real-time experiments, this experimental data, suggesting that even in the most charitable and generous of conditions, you're looking at collagen preservation of a max, at least at this point, until something else is proposed, perhaps, of 500,000 years. Is that right, Joel?
That's right.
Okay, so if that's the max, even in very generous conditions, then couldn't those conditions then that you're allowing for be unrealistic in the real world? Like how many opportunities are we going to have for that in the past?
For sure. So, for instance, we're just saying at this temperature, excluding all involvement of microorganisms in decay, excluding temperature fluctuations, which are not nice to proteins. Proteins do not like temperature fluctuations, excluding the involvement of, somebody mentioned, the pH. So we're doing this in a neutral aqueous environment. Proteins hate changes in pH. The structure of collagen depends on a nice, appropriate physiological pH. All proteins are like that. And you change the pH. So we're doing this in a neutral environment, close to physiological pH. But all of those things are not guaranteed in the natural setting. So in the natural setting, you've got changing temperatures. You've got changing of the groundwater pH. You've got changing of the presence of or absence of microorganisms. And those are all going to lower the survival of collagen. None of those will increase it.
Which means the 500,000 years is a best case scenario for them, so even in their best case scenario, it's not even close to 65 million years.
That's right. Now I can throw you, so here's an example of an unrealistic rescue device. We can keep lowering the temperature. If you go down to like negative 40 degrees Celsius, that collagen is going to last forever. Like literally, it's just going to go on and on and on. So, but nobody has proposed that. And the reason nobody has proposed that is because, well, bones don't fossilize at negative 40 degrees Celsius. These bones that are being found are, we can project that they were fossilized in approximately 10 degrees Celsius.
Yeah, I was going to ask if you tested any unrealistic scenarios just to kind of see, what might preserve these things longer. But you're saying -40, huh? I don't know where you're going to find that anyway.
Yeah. The ground temperature doesn't go -40. So that's an example though of one of those unrealistic preservation mechanisms. And another one, we need to test just kind of for fun to do that unrealistic. Test it with formaldehyde. As you know, formaldehyde is actually used to preserve things because it cross-links them. So we plan on testing it with formaldehyde to see that's an example of a very unrealistic scenario. If that does not preserve it, boy, then nothing will. But I do think something like that, as extreme as that, is going to extend its lifespan.
Yeah, you also mentioned that some people have tried to posit that there are exogenous tissues. What do they mean by that? They mean that they're contaminated, like a newer type of animal died in that same region. Can you explain that a little bit? Is that something taken seriously?
Yeah. Well, okay. So first, I'll tell you what they mean. They mean two things. One is contamination by the scientist or by the handlers.
Okay.
So when the bone is extracted, someone was eating a turkey sandwich nearby, and now there's this collagen that shows up in the samples. So that's one reason. One is contamination by microorganisms. This was primarily an attempt to explain soft tissue, that the soft tissue was like a microbial film which coated these things, maintaining their structure, but the actual tissue was long gone. That was the initial rescue device, actually. And you don't hear that anymore. The reason you don't hear that anymore is because, credit to Mary Schweitzer, she has gone above and beyond to demonstrate that there are actual vertebrate proteins inside of these dinosaur bones. So it could not have been microbial.
And the sample was large that you showed. I mean, it looked like a large piece of material that was stretching. It wasn't at a molecular level.
That's right. That's right. So there's more than people even anticipated.
Yeah.
Now, to your point, though, the paper that I shared, the other paper that did collagen decay stuff and measured kind of the ceiling, how long it could last, and we compared our data to theirs. They are some of the kind of remaining skeptics on this issue. They're still a little skeptical about that these proteins are actually dinosaur proteins, and the reason they're skeptical is because they are chemists that study the decay rate of these things, and they do not see how it could have survived 65 million years.
So therefore, is there any, because I've heard this argument used over the years, but I've noticed in reading articles and rebuttals from old Earth creationists, not even they think this is a good argument, but they'll say, well, these biomolecules could be the result of bacterial contamination, and that contamination would basically give the false appearance of blood vessels and cells. But with what you're saying, is there's not too many people that even look to that argument anymore, Joel.
But and here's why. That was a good argument when you were just looking at like the shape of tissues.
Okay.
The shape of the cell, because maybe the shape was preserved by the bacteria. Maybe the shape of the blood vessel was preserved by bacteria. But since then, in the last 20 years, now we have the technology and the ability to you can stain. How do I explain it simply? You can treat the samples with these antibodies, and you can see what proteins are there. And you can see that collagen is present. You can see that actin is present. Collagen is only in vertebrates. So there's no microorganisms that would have deposited that on these samples. And that was kind of the nail in the coffin of the biofilm hypothesis.
Do you think we've reached the point yet, or no? Maybe we need more testing, like you've been saying with the cross-linking argument. Has there been a nail in that coffin yet, or is that something that's still kind of being worked out?
Oh, yeah, that's. I'll admit that's going to be a tough one to put a nail in.
Yeah. Okay.
Yeah, because it's sufficiently ambiguous. like, oh, well, how do we know what conditions it actually was? And maybe the conditions you tested weren't representative of the conditions that would have preserved it. So it's sufficiently ambiguous to avoid falsifiability.
It's in the world of historical science where they could come up with a story and there's really no way to falsify it. If it's a story like that. So one thing I wanted to ask too, because this is so common, the assertion that we see from so many critics who might not be as up to date on this as they should be. But I think watching this presentation and a couple others that we've hosted will get people up to date. But they'll typically assert that, well, Mary Schweitzer, it always goes back to Mary Schweitzer and her findings from years and years ago, as if that's the only one. And then they'll say she's not even a young earth creationist. So it's not like she's concluding a young earth. Why would we? And we've heard all these points before. But in your presentation, you made the point that there are over 100 papers documenting the existence of these biomaterials. Is that right, Joel?
That's right.
So it's not just one, it's not just two. I mean, there's over 100 papers. So would it be accurate to say that this is becoming more and more widespread, more commonplace?
Absolutely. To the point where people don't really even give it much notice anymore. Like nobody publishes about soft tissue being found in this new specimen. No one even looks anymore because it's like, well, that's old news.
It's kind of like junk DNA,
Yeah.
How junk DNA, it really is junk science. They accidentally find these functional roles for so-called junk regions of the genome, ERV, pseudogenes, so on and so forth. And they find them by accident. So now that they actually look, they find more and more function. And it sounds like to me, they by accident found a lot of these biomaterials, right? These soft tissues. And then now that they've decided to, over time, look for them, now we're finding more and more because, oh, look, so it sounds like the evolutionary paradigm, Joel, hinders scientific advancement because they're not looking for these things and then they find them accidentally.
Yeah. The DNA is a perfect example. I can show you example after example of scientists who say dinosaur DNA does not exist. And so they're not going to look for it because it doesn't exist.
Start saving all those quotes.
Yeah.
It's like the shock of the fact, and you said you put it in at the end of your presentation, which was great. The Neanderthal DNA. Well, how come we have readable Neanderthal DNA and these so-called hominins that existed hundreds of thousands of years ago? It shouldn't be there. It should be degraded. You certainly shouldn't get any good data from it. That's for sure.
And let's not sugarcoat it. We've got our work cut out for us. This is a truly challenging task to get this ancient DNA. And I can't think of any example of flood deposited DNA that has been sequenced. So, we would it's not going to be as universal and as commonplace as proteins in soft tissue.
Right. And Neanderthals, most people agree, are post-flood. So that would be
and mammoths post-flood.
Okay, so here's a big one, and we've gotten a few questions, and it's right here in this topic that we're on. We've had several questions, and it's been a lively live chat, so I'm trying to keep up, and I'm trying to find this specific one. But so I'll just reiterate it as I'm trying to find it. Basically, okay, so we find, DNA in Neanderthals. We find some examples of soft tissue, red blood cells, collagen. But if the earth is young and the flood is true, why don't we find just massive amounts? How come we don't find DNA everywhere? How come we don't find DNA in other hominins like Erectus or Hobbit or so on and so forth? So in other words, they're kind of asserting that, well, if young earth creation is true, then we should be finding more of this. Joel, I'm curious your thoughts on that.
Yeah, it's a funny, flip of the argument.
Yeah, right. Yeah. Flip it on us.
So actually, you may have noticed I make a really big deal about the structure of collagen in my presentation and how it is an exceptionally stable molecule. And that is on purpose because we as creationists have to explain how this protein survived thousands of years. That's remarkable. That's an extremely long time. Let's not forget that thousands of years is an extremely long time. So when you go to something that's a lot less stable than collagen, such as DNA, The half-life of DNA is on the order of hundreds of years. So it's around 500 years is what I've heard reported. And so on the order of hundreds of years. So that's we would not expect, even from a young Earth perspective, we would not expect there to be just DNA in everything. And I wonder, now let's just think out loud, right? Let's just think out loud.
Yeah.
The flood preservation conditions may not have been conducive to a lot of really good preservation of DNA. That's something I'm privately concerned about, because here we are, we're going to go for this DNA. Dinosaurs basically died out in the flood, so the only chance of getting it would be from a flood laid down sediment, and it's evidently the flood was not kind to molecules such as DNA.
Sam, I'll say this, and then Sam, I'm gonna throw it to you, because I'm thinking out loud too, and I'm seeing, so right here, Reboot, appreciate that. That was basically an answer to his question. Dr. Joel Duff, appreciate you being here. He's a critic and evolutionist, and so he appreciates your prediction on finding DNA in flood fossils. And again, I'm just thinking out loud because of the Neanderthal DNA that we find. But in a lot of these examples of post-flood DNA, like Neanderthals, mammoths, these lived during the Ice Age. With Neanderthals, they lived across Eurasia. So those Ice Age related conditions might be more conducive for DNA preservation. But during the flood itself, and you kind of just hinted at that, would those flood conditions necessarily demand preserved DNA, do you think, Joel?
Yeah, I mean, it certainly would not be, it wouldn't favor the preservation of DNA. Cold temperatures is good for every molecule for long-term preservation. The flood was anything but that. So there would have been an extended period of elevated temperatures with all the volcanism, and then that was followed by the Ice Age. So the flood itself is kind of like a warm spell on planet Earth, and that doesn't spell good news for trying to find preserved DNA. But if you have a few isolated pockets where the DNA is fairly well preserved, getting it to last the remaining 400 to 500 years since the flood, that's doable,
right?
So I think from that perspective, we would not expect it to be universal, but we would expect it to be present.
Yes, I agree with that. I think that's a great point. Sam, over to you, brother.
Yeah, no, I was gonna say the same thing. I don't know why we would expect to find that anywhere, really.
Or at least in abundance.
Yeah,
like maybe a couple local, because really, as young Earth creationists, we don't have to fear it. We could find one example, and it's like, okay, that makes sense. But one example to me would be too much for the old Earther. You're finding DNA in a T-Rex or Spinosaurus or Mosasaur or whatever.
Now, for instance, in the 430,000-year-old hominin samples, those are extremely fragmented DNA. So let's say from a biblical perspective, that's post-flood, 4,000 years old, give or take. And the preservation is not great. Even at that, it took forever to have the technology and the resources to be able to sequence Neanderthal.
So apparently, and Sam, you were talking about this pre-show, apparently they're now finding remarkably well-preserved soft tissues in some trilobite fossils, including the mouth and digestive tract. Now, I still have to look into that a little more further to confirm, but Sam, I see a movie, if you want to elaborate on that. I mean, I'm curious the implications of that one.
Yeah, I did. It was brought up in another presentation on your channel in the recently, and so I just did a quick search on the internet, and yeah, it turns out that they are finding the soft tissues now in the trilobites, which would be a huge problem. But I don't know to the extent. I don't know if it's the same extent of the dinosaurs, but it seems like it would be. It seems like as we begin to continue to find those, I don't know why it would be a whole lot different. That would be another prediction, I suppose.
Yeah, I mean, look, this is a very risky prediction, and we shouldn't shy away from risky predictions, because that's where the most powerful predictions are. The risky prediction is that That geologic column does not represent time. It represents order
Right.
during the flood, and therefore they are all approximately the same age. I mean, and that is a huge risky prediction. When I say risky, I'm talking that from a scientific sense. Risky predictions are predictions that are very specific to a particular hypothesis. So the biblical worldview makes that very risky prediction that all of those are the same age, and therefore it should not surprise us that we could extend this back further.
It helps. Yeah. It helps demonstrate what we like to say, that it's not necessarily time, but a process. And the global flood allows for the necessary processes to rapidly form all these geologic features, fossils, soft tissue preservation, so on and so forth. And so here's a good question from another skeptic. Again, we appreciate our skeptics. We want to leave no stone unturned. And especially on this topic, because this is certainly a topic that should get open-minded people thinking. It truly is fascinating evidence here. And I'm looking forward to the future work too that you're doing and other people, Joel. And so Sydney Raptor is wondering, question for Dr. Brown. Not objecting to the science, thanks for that. But what is the direct, testable, positive evidence for the design aspect that you slip in with the good science?
Sorry. Sydney, if you're still out there, feel free to chime in the chat. By that, perhaps, Sydney, you're referring to like my claim that the collagen molecule is that triple helix is a beautiful design and designed to be stable because it's a structural protein. I'm not using that as, well, let me just be clear. I'm not using that as like proof in the scientific sense of the word. That is me thinking out loud from a biblical perspective. So from a biblical perspective, I believe, let's put it that way. I believe things look designed because they are designed. And I'm not using that as proof. I'm using that as that is what we would expect from a biblical perspective. So what is design? And maybe that's what you're getting at. Design is specific complexity. So complexity for a purpose. So that collagen molecule with that design of a glycine followed by a filler followed by a proline, that's complexity with a purpose. The glycine is the smallest amino acid, and it's the only one that fits into the turning of that helix. So that's a design component. It's complexity for a purpose. And the proline is an amino acid that has a kink. It's one of the only amino acids that has a restricted conformation. So that's actually what gives the collagen fiber its ability to form these tight helices. And so I look at that, I see a complex arrangement of amino acids. That's complexity. Complexity alone is not sufficient to call design, but complexity with a purpose or specific complexity. So that complex arrangement of amino acids also correlates with then a very specific function, in this case, stability.
That's a great response, because we like to focus here on these different complex mechanisms, these chicken and egg problems where, design is the best explanation for these interdependent systems that we can talk hours and hours about. But specifically when it comes to the soft tissue and the collagen and these biomaterial preservations, the predictions would be more so in the world of, I think, what you've been talking about, the flood, a young earth, looking for DNA in dinosaurs. That's where those predictions would be, rather than design, Joel.
Yes.
Yeah. Okay.
No, I just slip in the design stuff as freebies.
Yeah, which is
because
good.
I believe it's designed, and I'm not say so.
And we got no problem with that. We agree. Okay, so I got another question here from, actually, before I pull this one up, Sam, did you have a follow-up, brother?
I don't think I do. I think I had a few, but I think a lot of them are very similar, and I think that you've done a fantastic job of answering those.
You have, because especially the big ones like the cross-linking, the iron, which you're obviously familiar with, Joel. So I appreciate you helping us to leave no stone unturned. And so we got one here from CC, and he's wondering, occasionally biological materials don't break down. They can become sealed inside stone as the deposition process occurs. They need to be liberated by dissolving the mineral away. No DNA. I guess he's wondering why.
So why is there no DNA in those situations? So I don't know is the short answer. It's a good question, and I think let's speculate. So it's probably because the DNA itself is degrading pretty rapidly before it's sealed.
Yeah, that makes it well, and this goes to the future testable predictions aspect of the model too, right? Looking for DNA, or am I missing something on that one?
No.
Okay. All right, let me scan the chat here and see if, because I think we've done a pretty good job at addressing the majority of the objections. Sydney Raptor's wondering, in the very stable, see, a lot of these comments came in too during your presentation, so they're going to be related to certain aspects of what you were talking about at the time. And this was near the Mary Schweitzer part, but he's wondering, in the very stable rock strata that Schweitzer's material came from, ? Does that make sense, Joel?
Yeah, so we still know it's permeable enough for water. Water gets in there because that's how these things fossilize in the first place. It's the replacing of the mineralized content with the inorganic minerals. So water is still getting in these, even though the rock strata itself looks pretty impermeable.
I'm curious on another note, because we're talking a lot about collagen and proteins, and as we start to wind down here, one of the major arguments for common descent in general, which they'll look to the so-called geologic column, and they'll look to the lower layers followed by the middle to the top, and say we got a series of common descent here. But in the world of genetics and molecular genetics, you as a molecular biologist, they'll say, well, these similar proteins, and you'll see people like Richard Dawkins make this argument too, that similar DNA, similar proteins that we find in basically the entirety of the biological world, the fact that they fall out into nested hierarchical patterns, more similarity in proteins and DNA between humans and chimps than between humans and dogs or humans and fish, they'll say that suggests strongly common descent. And I'm curious, Joel, how you feel. Obviously, we have many different ways we could approach that as creationists, but how do you feel is the best way to approach that argument?
Yeah, okay. So I would like to pose the question, if you were designing it, how would you design it? And initially, I used to think that, well, a designer could have designed them to be like completely independent organisms with no shared sequence between them. The designer is not constrained to have designed that shared sequence between those two. But that's not true, actually. As I realized this going through my PhD work, why would God create in kind of a nested hierarchy? Because we do see that in creation. We are biologically speaking mammalian. Why would God create us to be mammalian and have all these shared similarities? And then it hit me one day. Okay, the reason why you have these shared similarities from a design perspective, well, number one, it's good design, so you reuse the design. But no, this is that's kind of old hat. This is what I realized. It's so that you can study a fruit fly to learn about Alzheimer's disease. You can study a mouse to learn about neural tube closure. You can study a yeast to learn about cancer. And so the reason you can study a monkey to learn about human viruses. The reason you would create nested hierarchy is because in a world where you want the humans, your people created in your image, to be able to understand and study the world, you have to build a platform of similarities between the other life forms out there.
That right there is an incredible answer, and I appreciate it, Joel, because it adds another level or degree to how we typically respond, that human engineers build in hierarchical patterns, and so why wouldn't God build hierarchically? But it adds another layer to that. It's a four-dimensional answer, because that makes a lot of sense when you really think about it. The fact that we can study monkeys, mice, fruit flies, and we can learn more about disease. We can develop various sophisticated mechanisms and health treatments and so on, and learn about viruses, like you said. So basically, if we didn't have those similarities, or those nested hierarchical patterns in the biological world didn't exist in genetics, we couldn't carry out those kinds of research projects.
Yeah, imagine, for instance, what's the only way you could learn about humans if we bore no similarity to other life forms?
By testing fellow humans.
Yeah, exactly. Does that sound like a good creation?
No, that sounds scary. That's, yeah, which right there is a great answer. So to this whole nested hierarchy argument, which many evolutionary biologists, like, again, Richard Dawkins, there's a video out there called "One Fact to Refute Creationism." And I always love when they say creationism and not evolutionism, but Richard Dawkins' answer is that the fact that we have all these DNA similarities in the biological world that followed into hierarchical patterns. But the answer you just provided demonstrates that a good designer would design this way for many good reasons.
Yeah, and let me give a shout out. Like, this is one of those like outstanding questions for creation biologists, is to understand the extent of true similarities due to common descent, because there is some, dogs, wolves, coyotes related by common descent, and to understand the limitations. So I recommend, if you are a biologist out there, definitely get involved with the Creation Research Society, because there are a lot of people that are trying to answer these questions from a biblical perspective, understanding where those lines of demarcation are within the hierarchy.
And it's fun for us as creationists, Joel, because we can explain the similarities due to common design and due to the necessity for research, like you said, medicines, viruses, so on and so forth. But we can also explain the differences. So I like to say it's the differences that make all the difference, because in those research projects that you're talking about, which we need to be active in, we can look to these genetic level discontinuities that we as a creationist would expect, that separates kinds, that the evolutionist can't really explain because the differences are too massive. And I think that's a good way to approach that field.
Yeah, well put. Can I borrow that? The differences in the differences?
There you go. The differences are in the differences, which we could talk about all day. Orphan genes, Y chromosome dissimilarity between humans and chimps. And we can explain that. We can explain both. Yeah. But the evolutionists, they can explain similarities and some differences. But if the differences are too massive, that's where I think they have the biggest challenge. Sam,
Collagen is a great example of that, because we see collagen, it's a great design, and it's used modularity throughout vertebrates. So it's used widespread. But then there is no equivalent in lower life forms. So it just pops out of nowhere. That's very hard to explain from a gradualistic evolutionary perspective. Whereas, like you point out, from a created perspective, we can explain the similarities and the novelties from a designer.
That's right. So you're saying the collagen, kind of like a Cambrian explosion,
yes.
Permian.
Yep.
Wow. So it pops up, no precursors, no pre-collagen, basically.
There are many types of collagen. Collagen type 1, type 2, type 1A, right. Type 2A. So there's a huge family of collagen in invertebrates, but there's no precursor to that.
Wow. That's incredible. Sam, did you have a follow-up, brother?
Yeah, just regarding the Dawkins question, I think that there's also theological reasons why, because God clearly created a world where that glorifies God and is evident everywhere. But at the same time, He created a world where people can suppress that truth and believe a lie. And I think that's what we see in this debate, because when someone alters their worldview, like we were talking about, then evidence of design is everywhere. And evidence of God is everywhere also. But as long as people are unwilling to believe and see it, well, then there's plenty out there for them to suppress that truth also.
Yeah, I guess it gives them enough reason to swallow the common descent pill. And, I also find it funny on the nested hierarchy argument. This is just a basic point, but it's not really a surprise that we share more in terms of genetics, Joel, with a chimp than we do with a banana plant. I mean, you could just stand afar, look at a human, a chimp, and a banana plant, and you could probably make that prediction ahead of time. We look more like a chimp. Anyways.
Yeah, it turns out our genes do have something to do with our appearance.
Right. Exactly. Yes. And so right here, we're getting a lot of love, brother, and a lot of positive feedback from our audience tonight. Time has flown by with you. I appreciate how comprehensive tonight has been. Wesley Coleman says, love the presentation. A big thanks to Joel Brown for his work. And I second that. Truly excellent work, Joel.
Well, thank you to everyone who's been listening in and commenting. It makes it fun.
Yes. Sam and I love the live shows because we get to interact with both critics and non-critics, and these kinds of questions help us to elaborate. And so I guess maybe the final question, just to really make this comprehensive, in terms of the similarity argument, Joel, and the nested hierarchy argument, the evolutionists will say, and I think there's a built-in assumption in the question and challenge, they'll say, okay, we understand similarities maybe due to common design. But what about regions of the genome that have no function, or they're simply evolutionary leftovers, they're junk? Why would those fall out into a hierarchy? Why would we have similarities there at all?
So two answers to that, or two responses to that. Number one is we would actually predict that every region for which there are differences and those differences are shared. So shared differences is functional. So we predict from a biblical creation perspective extreme function throughout the DNA. So what they are often calling shared mutations, we would predict that there's actually a functional consequence or a functional reason for that to be designed into there. So that's a risky prediction. Another good, strong, risky prediction by the biblical worldview. One of the reasons why junk DNA has really fit so much better, junk DNA, into the biblical story than the evolutionary story, because we predicted that all along. Although we really should have been more vocal. We need to be a little bolder about our predictions.
Yes.
Number two is, and this probably applies in some cases where there is a propensity for that region to for mutation. So you can have the same deleterious occurrences occur in different unrelated organisms. I think
a mutational hotspot, basically.
Yeah. The vitamin C glucosinoltransferase is probably an example of that, where you have loss of vitamin C synthesis production in multiple organisms, including humans, guinea pigs, some bats. It occurs throughout.
So, and I think that's a great answer. Some of these so-called mutations are true mutations. Let's take the gulo pseudogene, for example, the loss of vitamin C synthesis, while others, if I understood your first point, are created differences. They're created for functional purposes, because it's the evolutionist that has that assumption that all DNA differences in DNA variety, right, Joel, are the result of mutations over time. But we as a creationist, we need to differ between created differences, created heterozygosity, the technical term for it, and true mutation since the flood.
And here's a great example. Do any of you have brown eyes? Sam, it looks like you have brown eyes.
Blue eyes.
Blue?
Yeah.
Donnie?
Blue.
Oh, man.
Yeah.
So we're swimming in the shallow end of the gene pool. Blue eye color, it has many genetic influences, but it can be traced to at least one major influential location in the genome, which is the difference of a single base pair. A single base pair.
Wow.
That changing that one base pair influences the expression of genes nearby and influences the amount of pigment that builds up. In other words, something that others, like an evolutionary secular perspective, would call an insignificant base pair. Well, it turns out it's a binding site that regulates nearby genes. And I think that is not the exception. That is the norm.
Wow. That's incredible, Joel. And just a lot of research opportunity there. That's why I like to say it is a great time to be a biblical creationist, because there's so much there in terms of making predictions. And the fun part testing those predictions, not just making them, but actually testing them like you do. And I agree with you. As creationists, we need to be, as you have been, we need to be bold about those predictions. I think that showcases the power of the biblical creation position. So, Dr. Joel Brown, I've really enjoyed this. 2 hours has flown by. Perhaps at some point in the future, we'd love to do just a show, maybe throwing questions, general questions on evolution at you, because you have such great answers here. And so, Joel, I do want to hand it over to you in case you had any final words, final thoughts, essentially anything you wanted to leave Sam and I, and also our audience with tonight.
Well, hey, let me start by just saying thank you. I've really enjoyed this time. This is my first with you all, and I really appreciate what y'all are doing. I want to just finish by always pointing it back to the creator. This is fun. This is interesting, and I would do it just for the fun of it. But this is ultimately about giving credit to whom credit is due, because we serve an amazing God, creator God, who actually cared about his creation enough to die for it. And therein is the gospel. And that is where I would just want to finish by pointing people back to is the loving sacrificial gift of God's Son, Jesus Christ, and invite you, if you are not already there, invite you to have a relationship with that Savior.
Amen. Those
Amen.
are the best final words
Right.
and final thoughts possible. An appeal to the gospel. It all comes back to the gospel. Winning souls. Again, we could win arguments all day, but it's about winning souls. Joel, I've really appreciated your time here tonight. I've been really looking forward to this, and you did not let us down, brother. And so we've got the links here in the live chat. Check out the Creation Research Society, creationresearch.org. And you got a lot of great content there. I've also got the links in the description box as well. And you can support CRS here. There's the link, and looks like students get a free membership. So that's cool. And yeah, I hope people check that out. You guys are doing awesome work. Clearly, we got a glimpse tonight in terms of what you guys are doing with research. And I love how you always bring it back to let's study it. Let's experiment. Let's get some real experimental data here, Joel. And of course, Sam, our award-winning co-host, brother, thank you for joining us for tonight as well. Did you have any final thoughts for tonight?
Dr. Brown, thank you for your time. It's been a pleasure. And Donnie, thank you for having me. It's always a joy and a blessing to be able to just come and be a part of these conversations with you.
Hey, it's my pleasure. We got a lot of people in the chat saying, can't wait to watch this. Cam says, missed it, bummer. Don't worry. It's going to be available as soon as we end it here. And I really do want to encourage anybody who might be just joining us here in the live chat, do make sure to watch it right from the beginning. Joel, you gave a fantastic presentation. And in our Q&A and discussion here, we really did our best to leave no stone unturned and give you a comprehensive program on what? On soft tissue and molecular biology from a creationist perspective. I hope you all had as much fun as I did. Again, Dr. Joel Brown, really appreciate your time.
Thank you.
And Sam, thanks so much for co-hosting tonight. God bless you all. Standing for Truth is out.